Thanks for surviving up until the very last talk. Uh, hopefully, hopefully you won't regret it. Um, I'm currently at Indiana University, but in 2 weeks, I'm hoping that I'll be in NCI, the National Cancer Institute in Bethesda, Maryland. We have a fantastic new lab focusing on cancer data science. And we are recruiting postdocs, PhD students, and hopefully investigators, faculty. So keep us in mind. I'm not a GWAS person at all. Any mistake that I'll make, I apologize. Correct me whenever you feel like. So this is sort of a new experiment for me. However, I do care about algorithms and I do care about resource-bounded computation, and sketching is one way to address memory limitations. You can have a grasp of how sketching can be used, especially for secure genomic data analysis in the context of Intel SGX. So obviously, we are dealing with larger and larger data sets, and there are many inference tools based on statistical machine learning that need to integrate a lot of omics data. But they need large-scale, multidimensional data from diverse populations, and individual institutions cannot always have the right scale of data to perform meaningful inference. However, patient data across institutions cannot be shared across institutions due to privacy concerns, especially when multiple countries are involved. I mean, you all are facing this. You want a piece of data, especially when it's related to electronic health. They can't be shared. So what do we do? As especially demand for rare disease studies increase, we have to find a way to preserve privacy while performing statistical inference on shared data. Perhaps we don't have to see each other's data explicitly. Perhaps a third party can perform computation for us in the following manner. So we'd like to do some hassle-free and efficient data sharing that involve multiple research groups. They don't even have to be research groups. We'd like to do it in clinical practice too, while maintaining each party's data privacy. And eventually, once the computation is performed, summary statistics need to be shared across all parties. So there are multiple emerging approaches for this. You may have heard secure multi-party computation, homomorphic encryption, and so on. But the solution that I'm going to present today is based on what is called a trusted execution environment. How many of you are familiar with Intel's SGX Secure Guard Extensions platform? Okay, not so great. Maybe it's time to learn about it, because every Intel processor, generation 6 and above, offer this, and you can make use of it. So all the cloud platforms that you're using, they're consisting of processors that provide you this possibility. And yeah. Let me give a brief description of what SGX entails. So it's a private region of memory that is called an enclave. The contents in the enclave are protected. It cannot be read or saved by any process outside the enclave itself, including the operating system. And the enclave is trusted, but the processes outside of the enclave are not, such as, again, the operating system. So it can be run in an untrusted cloud environment. The hardware requirements are minimal. I'm sure that pretty much everyone in this room are using Intel processors that support SGX. There are multiple enclaves permitted per processor. The only downside is the memory that you can reserve is pretty limited, 128 megabytes in total for all enclaves that you can have in a processor. So here's a general workflow if you'd like to do GWAS in SGX. Take each and every VCF file that you'd like to compute with. First, you have to do some simple segmentation. Then you better perform some compression because VCF files are very wasteful, as you know. Since you have limited memory, compression is essential. Then you have to perform encryption. So across all parties collaborating, you have to set up a common encryption scheme. Then the encrypted files are transmitted to the third party. Again, it can be untrusted as long as it supported SGX. Obviously, within the enclave, the files will be decrypted, decompressed, and then some joint analysis will be performed. And based on the protocol that the collaborating parties have, the summary statistics will be communicated back to the users. So it looks pretty trivial. I played a minor role in a study on Kawasaki Disease. It's a rare childhood condition that involved 3 international parties in the US, UK, and Singapore. And these folks demonstrated how to use this general workflow efficiently. So as long as the number of SNPs that you deal with is reasonable in number, you don't have to do anything beyond what I already showed you. Based on the success of this study, some of my colleagues, Haishu Tang and Xiaofeng Wang and others, who organize the iDash competition every year. How many of you are familiar with iDash? This is a fantastic competition for secure computation, especially when it comes to genomics and bioinformatics. I encourage you to consider attending. Every year they have a couple challenges, and in 2017, one of the challenges was generalizing this. They asked how we perform secure GWAS, how to compute the chi-square statistic in a given bunch of VCF files, including both Ks and control samples, in an SGX environment. It was simple enough. So the number of SNPs altogether were about 60 million per VCF file. There were 1,000 cases and 1,000 controls. And the idea was to figure out the top 10 SNPs associated with a phenotype vector. I hope the challenge is clear enough. So we participated, although I'm not a GWAS person. I like compression, and we had a couple decent ideas. And the implementation was pretty straightforward. With respect to speed, unfortunately, we didn't win. With respect to compression, we were by far the best. But I argue that even in this first version of our tool, we were scalable and we were generalizable. And it was a good starting point. So we didn't do anything far beyond what I already mentioned. With the only exception being in compression. So the most simple way of performing VCF file compression is you take the SNPs, you reorder them based on their IDs. You make sure that you separate the SNPs based on zygosity, so you don't have to keep all these additional columns. And then you perform run-length encoding, you differential encoding in subsequent SNP IDs. So you can reduce the size of a VCF file quite a bit like that. And then you have to perform the chi-square test. How many of you are familiar with the chi-square test here? For those of you who are not super familiar, the easiest way to think about it is, suppose that, as I mentioned, R is the number of cases and S is the number of controls. if they are equal and they are n over 2, half the size of your data set. And thinking that here, Ri0, for example, indicates the number of 0, 1, and 2 indicates the number of minor alleles. It indicates the number of cases with homozygous major allele, right? And 2 indicates those with homozygous minor allele. And in case overall, in sum, the major and minor alleles are half and half, what you'll get is basically the ratio between the minor alleles in cases versus control. So that's all there is about the Chisquare test. And it's pretty easy to compute if you don't have any memory limitations. So the key question is, when you go up to the entire human genome, can you fit everything in the secure enclave? The competition itself focused on chromosome 1 only. That is 1/12 of the human genome. When you generalize it to the entire human genome, the space was not sufficient. So what we need to do is come up with a way to identify the top SNPs, top 10, 100, you name it, with limited memory, where you cannot have a single memory unit per SNP. You have to be a little more innovative than that. So the original solution that we had was using a standard hash table, even though there are some subtleties there. For example, if you're familiar with Robinhood hash table, that seems to do better than others. The real challenge was coming up with a sketching system that enables you to identify the top SNPs. In addition to this, we also wanted to support online querying. So assume a scenario where the VCF files are all in the cloud. Their phenotypes from a phenotype vector is available. You'd like to recruit some of these files for your statistical test for a particular phenotype or a combination of phenotypes. And you should be able to perform these tests after specifying which VCF files you'd like to be able to use. And all you're going to get as a result will be, again, the IDs of the top SNPs. And our system supports that. So how do we do this by sketching? I'm sure that you have heard about streaming and sketching methods. From a theoretical computer science point of view, sketching has a specific meaning. And I'll try to illustrate it to a degree. Consider the following value, Fi, for a given SNP is basically the allele frequency difference, especially thinking if S would be equal to R, simply you're calculating the allele frequency difference. It turns out that this simple value with respect to SNP ranks closely resembles the chi-square value. So all I'm going to say is, rather than first ranking the SNPs based on the chi-squared values, I'm going to rank them according to the F values. And I'm going to not only keep the top K SNPs according to the F values, I'm going to have some L SNPs according to this, which I'll show is relatively easy to do. And the L value will be, say, for 100 SNPs, I may be interested in figuring out the top 100,000 SNPs. And I'll guarantee with high probability that every single one of the top 100 SNPs, according to the chi-square statistic, will be among those in the top 100,000. So hopefully the general scheme is pretty clear. What we have is a false negative free proxy for the chi-square statistic. Yeah?
But you're essentially using the same information that
Yeah. It's pretty much identical. Yes, but this what I have is linear. And I can use a linear sketch. which can be implemented as follows. So how many of you are familiar with count min sketch? A few. So I'll go through count min sketch. First, I'm going to calculate the allele frequencies for the cases and controls separately. So it is additive. Let's focus on the cases. I'm just going to come up with a count of a particular SNP in a space much smaller than the number of SNPs. How do I do that? I'll have multiple hash tables given here. Every row is a hash table. And the number of columns is, say, 100 of the number of SNPs. And for each hash table, each SNP has an entry. I will update the entry for each hash table. And in the end, the representative for the particular SNP, once I'm done with every single SNP, every single VCF file, will be the minimum value. The probability of that representing the true value of a SNP with a high count will be very high. And this is the whole idea. Does this make sense? So this is the count min sketch. There are some additional linear sketches, such as count sketch. I'm not going to get into this. I just want to give you a flavor of what can be done. The key idea is by doing this simple computation, I can have a good estimate on the particular SNP's case count as well as the control count and the difference as well. You can prove that you have a good approximation for the difference. And the top L SNPs, once again, L should be maybe 1,000 times that of K, would include the K SNPs that you may be interested. So this was already an achievement, but we didn't stop there. One of the major challenges in GWAS is obviously PopStrat. How many of you are familiar with population stratification? So we had to handle population stratification in the secure enclave as well with small memory. So our main challenge is everything has to fit in this 128 megabyte limited memory. And population stratification corresponds to the following phenomenon. So this is both a research talk and an overview, if that's okay.
It's like your interpretation of our field.
Right, exactly. So it's a naive person's understanding of years of GWAS. So that was the message we were able to deliver to the population. You know? So here's a cartoon picture of PopStrat. Suppose that you have two populations, the top population and 1 and population 2, and suppose that the reds indicate the minor alleles and the blues are the major. If there's a clear difference between the distribution of the minor alleles among the two populations, and by some luck, the cases that you picked are heavy on population 1 and the controls are heavy on population 2, you'll observe a spurious association. So this is my cartoon interpretation of PopStrat. You'd like to avoid this. So one way to do that is through an Eigenstrat strategy. Again, I'm wondering how many of you are familiar with Eigenstrat. Some. Again, I mean, here is the way I think about it. I'm sure that it's a very naive way of thinking it, but it helps me understand. So if you look at the picture on the left, the A vector, the genotype vector we have SNPs on the rows and samples on the columns. And you'd like to figure out the principal component, first principal component of the A transpose A, the matrix that correlates samples with others. So the top, the first principal component that you may get is indicated by the axis of variation. So it tells you basically how much in agreement or how much of difference each sample would have with the rest. And suppose that the candidate SNP that you'd like to check for association with the phenotype, which is also given next, is indicated there. By a naive look, you may think that the candidate SNP is strongly associated with the phenotype, but when you perform a correction with the PCA, which means you take an orthogonal mapping of the candidate vector, candidate SNP vector, and the phenotype vector with the first principal component, the vectors are given next to those, you see that the association pretty much disappears. So it basically corrects it for the differences within your sample set. Unfortunately, this cannot easily be done in a small memory environment. So what we have done is, I'm not going to perhaps get into the details of a particular algorithm that we have used. Basically, A is the matrix that we have been using. We have to perform first some mean centering. And then we are going to use a linear sketch S. And need to perform the singular value decomposition. We remove the principal components and perform all the computation on what we get. And we can provide additional guarantees on the eventual matrix that we'll get, such as we are closely representing the original matrix X after population stratification with our resulting matrix. So the details can be found in the stock paper from 2016 by our colleague David Woodruff and colleagues. An additional contribution that we have is going beyond the standard chi-square statistic, and we check whether we could do Armitage-Corcoran trend test, which is closely related to chi-square statistic. And it turns out that you can do reasonably well in both. Again, I don't think that there's a point in getting into details. So in the end, our goal is taking a genotype matrix, perform pop-strat correction, then perform an association test, and return the top K SNPs with respect to Armitage-Corcoran statistic. I'm not going to, again, get into some of these details, but basically, the bottom line is the linear sketch can be applied to the transform matrices, and the top L SNPs that we identify will include, with high probability, each one of the top K SNPs according to the statistic of choice. So I'm wondering if the general idea is reasonably clear. So, yeah.
So maybe I'm confused, so maybe others are confused.
Mm-hmm.
So what's the exact intuition behind your, like, I mean, so you're loading the whole matrix into, you can't load the whole matrix into the...
Yeah, we can't.
You can't, right?
So we are keeping a linear sketch of it.
Sketch of the whole matrix.
We have a linear sketch of the whole matrix.
Okay. And that's, okay. And then you have some theorem that says that that's going to preserve the...
The reason is the standout SNPs will have high counts. So think of, again, count min sketch example.
Okay.
Since you are adding up multiple SNPs in each hash table, what is the probability that each one of these hash tables will penalize a particular SNP of high value, which is among the top K.
I see, but it's not going to work, for example, if one site contributes all the cases and the other site contributes all the controls.
What do you mean by one site contributes?
Like, all the cases come from one institution and all the controls come from another institution, because then you won't be able to...
No. Each VCF file, each individual is encrypted separately.
But it has all the cases and the controls for that site, right?
No, not necessarily. I don't care about where each one of these individuals come from. I'm going to treat them separately, whether they are from the same institution or not. For me, they're different. Each VCF file is processed independently. They contribute to the hash values, and then I'm done. I move to the next VCF file. Whether it's from the same institution or not, I don't care.
Okay. I see, but you're only keeping like the top set, right? Not everything.
Yeah, exactly. So the guarantee is only for those SNPs whose counts for both case and control is significantly different from the rest. So suppose that there are some SNPs that I care about that are highly represented, and there are those that are not represented. In case or control, it doesn't matter. So the question is, in these hash tables, in how many of these hash tables can you really make a mistake in aggregating two important SNPs together in that entry? And if you have sufficient number of hash tables, at least one of them will survive. Some of them will be conflict-free. Yeah?
It's one common sketch for VCF.
It's one sketch for the entire data set. The VCF files, if they were provided to me row by row, my life would be very easy. But they're provided to me column by column. And every time a column comes, I update the sketches, which is, in a way, a random sum of multiple SNPs. Every entry in my hash table is the sum of multiple SNPs. Right? I know which SNPs they are. I may drive them. But, oops, sorry. The question is, I am not sure what's happening. Oh, it's back. Good. Is the intuition clear?
I mean...
So suppose that there are 10 SNPs of interest. Okay. The rest are just unimportant. Yeah. Okay? I would like to make sure that, suppose that there are 4 hash tables. I'd like to make sure that one of these hash tables have, for each SNP, one of these hash tables guarantee that that SNP is not hashing to the same entry as the other important SNPs. That's all I care. The rest will not make a big difference. Okay. That's the general idea.
That part makes sense. What I'm trying to figure out is if you lump the cases and the controls together, how do you tell?
Right. Cases and controls are separate.
So you have common sketch for cases.
Exactly. If that's what you were asking, I'm sorry, I didn't get it. Okay. And then I can do the difference, and you can still prove that the difference will be close to what it's supposed to be. Okay, so let's give some numbers. With respect to compression, as I mentioned, if you're careful, you can reduce the size of a VCF file for our purposes by a factor of 50. So you only need 2% of the original data size. With respect to running time, you get the real differences. So with standard, if you have the memory for it, suppose that you focus only on chromosome 1, the running time for the I- competition data set was basically a couple minutes with a query time of less than 1 second. So that's pretty impressive. You have to basically there's a two-step process, right? The first step computes the most relevant SNPs, most relevant, say, 100,000 SNPs. There must be a second step in which you have to explicitly compute the chi-square values for these SNPs. And then you have enough memory. And within the memory, you can calculate explicitly who the top 10 SNPs may be. And that takes very little time. The update time, the hash preparation time may take a bit. When you use the count sketch is slightly different from the count min sketch, but you generally get the idea. It's a linear sketch with similar properties. This time the query time is a little higher, but you are given the option to process the entire human genome, so all 60 million SNPs can be processed. And with a hash table of size 2 million, again, remember that we are dealing with 60 million SNPs. There are 8 copies of the hash table. Still, there's a substantial difference between the memory needs and what you provide. The query time looks reasonable, and we have improved it quite a bit. Maybe you don't see it, but there's a dot between 55 and 32. So in less than a minute, you can perform querying, and the overall update time is by and large comparable to even with the original chromosome 1 dataset. So how about our accuracy? I think this is the biggest challenge that we faced. So here, let me try to summarize it for you. You see the ranks of the top K SNPs according to the Cochrane-Armitage statistic. And the y-axis shows what proportion of them were among the 2 to the power 17 SNPs identified by the F value, right? So up until roughly 700 SNPs were included in the top 2 to the power 17 that we identified by F. And this is, again, you have to look at the sizes of the sketches. The blue line shows the case where there are 13 hash tables, each with 0.5 million entries. And the yellow line indicates a much smaller sketch. There are just 8 hash tables, each with 0.25 million entries. So even with the yellow, the smaller case, you can reliably get the top 600-700 SNPs. With the blue line, you can see that the accuracy is perfect up until and beyond the top 2,000 SNPs. So the next shows, let me see. Yeah.
2 to 17 is significantly bigger than 2,000.
Yeah.
Right. So
it's about 128,000. But that's what we are suggesting. As long as that fits in my main memory, the 128 meg memory, I'm fine. And that's why it's 2 to the power 17.
So can I ask like
Yeah.
So what if you did the following? What if you just, took the first 100,000 SNPs, like load them into memory,
Yeah.
computed that for all the samples,
Yep.
and then did the next one and the next one.
Yeah, you can easily do that.
Perfect result. It just
Yeah.
be slow, right?
It will be just slow. This is only about performance, nothing else.
I see. Okay, so the idea is that the tuition is that you have a hash table, so you're kind of able to just scan through the data once and then capture all the.
That's right.
Okay.
Don't forget that every time you get a VCF file in the enclave, you have to decrypt it and decompress it. It's a lot of time. So again, with respect to accuracy, we lose accuracy First to pop-strat correction. Second to the use of F instead of the statistic that we are interested. And for both, actually, the accuracy is not too bad. By and large, we guarantee that the 2 to the power 17 SNPs that we keep capture the hundreds of SNPs, up to maybe 2,000 SNPs that you may be interested. We compared our protocol with secure multi-party computation protocol by Bonnie, Berger, and students. And as you know, SMC is considered a viable alternative to SGX. Some people have concerns about using an Intel-based platform. But when it comes to running time, there's simply no comparison. We are at least 100 times faster, if not more. So when it comes to performance, I think SGX is the choice. And you can see the communication is just not comparable. With respect to the I-dash data set, only on chromosome 1, the total amount of communication has to be over 1 terabyte with the SMC. And you can see what we have. It's just a few gigabytes. Anyway, so this summarizes our SGX-based implementation of secure GWAS with near-perfect accuracy that can handle large data sets, which performs population stratification correction. There are several pieces of work ongoing, like handling linkage disequilibrium. Obviously, there's some normalization issues. So far, I've shown you what we have done with respect to mean, handling the mean. I haven't shown you anything about normalization. Adaptive queries, database persistence, and so on are other questions that we worry about, and more. With that, I thank my collaborators, especially David Woodruff and Oğuzhan Külekçi, respectively helping us with sketching and data compression. And the real two heroes are Can Koçkan and Kayuan Zhu, who came up with the entire set of algorithms, implemented them, and tested them. And thanks for staying up until this late.